Hello, everyone. Good morning. Welcome. And a special welcome to all our beloved mothers out there. HGP believes in freedom of speech, but the views expressed, especially by myself, our reader, today's speaker, HGP members, and our guests are not the official views of the humanists of Greater Portland. Humanism is a non-theistic philosophy which affirms the inherent dignity and worth of every human being, encourages the use of science, reason, and free inquiry, and promotes ethical principles and social compassion to enhance the quality of life. This morning, our reader is Ann Henderson.
I had a difficult time figuring out which reading I had 3 readings, and one was on travel. By the way, I'm wearing purple because was my mother's favorite color. And I was going to do travel because that's something that was my mother and I, have in common. And then I was going to do something, a Garrison Keillor thing. I said, you know what? I am the mother to my dogs. So I'm going to give you a poem about my dogs. Yeah, I know. When I am old, a dog lover's poem. When I'm old, I shall wear turquoise and soft gray sweatshirts and a bandana over my silver hair, and I shall spend my Social Security checks on sweet weed and my dogs, and sit in my house on my well-worn chair and listen to my dogs breathing. I will sneak out in the middle of a warm summer night and take my dogs for a run if my old bones will allow, and when people come to call. There am I. And when people come to call, I will smile and nod as I show them my dogs and talk of them and about them. The ones so beloved of the past and the ones so beloved of today. I still will work hard cleaning after them and mopping and feeding them and whispering their names in a sweet, loving way. I will wear the gleaming sweat on my throat like a jewel, and I will be an embarrassment to all and my family who have not yet found the peace in their being free to have dogs as their best friends. These friends who always wait at any hour for your footfall and eagerly jump to their feet out of a sound sleep to greet you as if you were God, with warm eyes full of adoring love and hope that you will stay and hug their big, strong necks. That's my Teddy. And kiss their dear, sweet heads and whisper to them of your love and the beautiful pleasure of their very special company. I look in the mirror and see I'm getting old. This is the kind of woman that I am and have always been. Loving dogs is easy. They are part of me. Accept me for who I am. My dogs appreciate my presence in their lives. When I am old, this will be important to me. You will understand too when you are old. And if you have dogs, to love you too.
Thank you, Ann. Now I'd like to introduce our speaker. He's a professor of biology and biological sciences at the University of Northern Colorado. His lab investigates fundamental questions in biology using venomous snakes and their venoms as a basic platform. He's received awards in recognition of his outstanding research and teaching, earning a BA and MA in biology at UC Santa Barbara and a Ph.D. from Washington State University. Please welcome, I lost the first name, Dr. Mackessy.
Right. Well, thank you very much. It's all right. My father says that Mackessy is the Irish pronunciation, not the Scottish pronunciation of Mackessy. So
Okay.
Irish. And he was a Chicago Democrat who did his best to vote early and vote often. So anyhow, it's a pleasure to be here today with you. As Pete mentioned, I am from the University of Northern Colorado in Greeley, Colorado, which is about an hour north of Denver. We're out on the plains, so the mountains are a good hour away, 2 hours to get up into Rocky Mountains. So anyhow, we have done a lot of different projects involving venomous snakes and their venoms. This is a eyelash viper from Costa Rica, and if you've been to Costa Rica, there's many, many natural areas where we have a lot of interesting flora and fauna. So it's a great place to see. All right, so we'll just, by way of introduction, what I'd like to do is first tell you a little bit about snakes, tell you a bit about snake bite because people are interested in that. And of course, that's where the primary interest and original interest in venoms came from, is when you're envenomated, it can be a very, very serious or potentially lethal scenario. We'll look a little bit at snake diversity globally and locally, and then talk a little bit about their biology. And then we'll get into venoms and what venoms are, how we go about studying these venoms, because we use a variety of different types of tools. We'll talk a little bit about how venoms evolved, and then I'll give you some examples of specific research projects, some of which are finished and some of which are ongoing. We've been interested in how snakes actually store venom, so we'll talk about that a little bit. Turns out that many different species of rattlesnakes and other vipers, and for that matter, other venomous snakes, show regional variation in their venom composition. So the biochemistry is actually different depending on where you are geographically. And then they show a number of adaptations for doing this, and that is feeding without limbs. And if you think about it, trying to take down something that's not particularly nice, a rodent has very sharp razor-like teeth, can be a challenge. And so vipers in particular have come up with a solution to that that's quite interesting. Finally, then we'll start talking about this concept of toxins to therapeutics, and that is, why are venoms a good source of molecules that may actually have and actually do have therapeutic value? So at the end, then I'll try to summarize this, give you some conclusions and take-home points. Okay. So can you see this? Are these titles clear up here?
Yes, very much.
Okay, because I've got a screen sharing icon in mind. So what we do in my lab is work with venomous snakes on a lot of different levels. And primarily what we'll talk about today are work with snake venoms, asking some basic questions. What is found in venoms? How did they evolve? How do they vary from one individual to another? How do they produce this cacophony of toxins? And in fact, I'll have it ready to go at a moment's notice, but don't envenomate or intoxicate themselves. Okay. Then we have done a number of different studies looking at, can we take purified venom components and use these therapeutically? And so we'll talk a little bit about that as well. Most recently, we've worked with a couple of groups nationally and internationally that are developing very interesting and novel ways to treat snake bites. So we'll finish up talking a little bit about that. Should mention also that we've done a lot of work with genomics, and this is looking at the actual entire genetic makeup of snakes and asking a wide variety of different questions. This is in collaboration with colleagues at the University of Texas, Arlington, who are genomic wizards, and it's a whole other interesting group of questions that one can ask and answers we can come up with using those techniques. We still work with the actual whole animal in the wild, and we've had surveys of Colorado that are funded by Division of Wildlife or Colorado Parks and Wildlife. We have done radiotelemetry work with a little snake called the desert Massasauga that's found in the southeastern part of the state. And currently we have ongoing projects with the natural history of prairie rattlesnakes that are very widely distributed across the plains from Mexico to Canada, as well as a more regionally localized species, the midget-faded rattlesnake, which is found on the western slope of Colorado and into eastern Utah, and basically in the Red Rock Desert country. So I get this question all the time: why do we study venomous snakes? Why don't you just kill them? Well, if you think about it, there's some pretty interesting things going on with these animals. They are adapted for living in a very harsh environment. And if you've been out on the prairie in February, for example, it's challenging to see how humans could make a living out in that kind of environment. Snakes have a good sense of vision in general, but rattlesnakes also have a well-developed sense of smell. They also have these pit organs here, a little hard to see in this picture, which allow them to sense infrared radiation. And so that's a pretty cool adaptation as well. And then finally, the tongue, which is forked, acts as a chemoreceptor, and they can pick up scents of mice and things like that in a very exquisite sort of fashion. We've done some work on how they're doing that and how they do things like relocate prey. Well, a lot of our work, though, has been involved in the material that's produced in this area, and that is the venom gland of the rattlesnakes. And then, of course, it's delivered via these. And the teeth of rattlesnakes are very much like a syringe needle. They have a bevel, they have a sharp tip, and they are replaced on a regular basis as well. So all sorts of interesting adaptations that these animals have. If you think about an animal that's only about 3 inches tall, living on the prairie and being able to migrate out and then return back to the exact same place year after year, it's pretty well adapted animal for living in that challenging environment. The other thing is that the number of people working with venomous snakes globally is rather small compared to other disciplines, but it's very, very international. And so we've had a chance to go to Costa Rica, work with a variety of different people and venoms there. Worked in Singapore with a colleague there on a number of different projects as well. And if you've never been to Singapore, it is a contrast of extremes. And so you have bizarre things like electric trees that anywhere else in the world will be tacky, but in Singapore, they're really quite cool. And then you have natural areas where we can see king cobras, crocodiles, and other animals like that. And that includes some of these animals. One of my favorite places on the north part of Singapore, and that's Sungai Buloh Mangrove Swamp Reserve. And so we can see these vipers there. There are monitor lizards that if you've never had a lizard that's 7 feet long, kind of lift up like this and consider you, it's a rather interesting deal. There are weird mammals, flying lemurs that occur in this area as well. So pretty interesting place and very, very diverse. I've also had the opportunity to work with colleagues in India and travel in India a couple of different times. And again, a very diverse fauna and flora for that matter in that part of the world, as well as a very long history and some really tremendous architectural feats. So if we think about snakes, though, many people fear snakes, and globally, that's kind of a universal human condition. And we've wondered why that's the case generally. And some of this is for good reason. Venomous snakes can, in fact, put you down, make you lose a limb or something like that. So, rather than try to decide which are venomous, which are harmless, just kill them all and let it go at that. This seems to be culturally transmitted information. And like a lot of other types of animals that represent a risk, humans typically lash out and kill them whenever they can. There's been some recent work done suggesting that there may be an innate component to this as well. And that's based on what we see with certain types of apes and monkeys and their responses, as well as other animals and non-human animals. So a colleague has suggested that venomous snakes and humans co-evolved in Africa. And so this aspect of avoidance or basically fear is an adaptive strategy. If you stay away from snakes, they're not going to kill you. Okay. Well, if we fast forward into the modern world, the reason for that is harder to justify. And some people are absolutely terrified. I remember bringing a snake into a classroom one time, and a student just freaked out and had to leave in a terror. It was in a cage, by the way. For one reason, snakes basically don't have enough legs. Spiders have too many legs, and those seem to be the extremes of digits that really set people off for one reason or another. But then there are others of us who find snakes and spiders and other things like that fascinating animals. Well, if we look at snakebite has been classified recently as a disease of poverty, and it's a neglected tropical disease. And the reason for that is that it differentially affects people in certain parts of the world more so than others. In Asia, many people are envenomated every year. In Africa and the Middle East, similar sort of thing. And in many of these places, it's challenging to get the treatment needed for these people because they live in very rural areas. Okay. The effects of a non-lethal bite can still be very, very debilitating. And this is a bite from something like this snake here, Terciopelo or Fertilance, a viper that's found from southern Mexico down into South America. Snakebite is a challenging problem to deal with medically because it's not just one toxin, but instead we have a whole bunch of different types of toxins that are targeting a whole bunch of different sites in the body of their prey and by extension, our bodies as well. And so we'll talk about a little bit about that in a bit. Well, World Health Organization has come up with strategies to try to reduce the snakebite problem by about 50% by 2030. We're already at 2025, and we're not really looking like we're on track to be hitting that target, but we'll still give it a try. As I mentioned, it's a very complex problem to deal with, and it's continuously evolving. So when you go to the hospital with a rattlesnake bite, for example, things are going to get worse before they get better, even if you get the appropriate treatment that's available. And so this can be very challenging for some physicians to try to deal with. The reason for that is the venom itself is a very complex mixture of a variety of different components. And again, we'll talk about some of the major ones that are found in the venom. Well, again, worldwide, rural areas are most problematic because it takes a while to get to proper treatment, if treatment is in fact available. In some cases, the venoms work so rapidly that it's not even an option, and the people die locally. So there are many, many challenges with human envenomations, and there are some pretty interesting new ways of trying to treat that. So, well, let's look at the snakes themselves for a moment. They're vertebrate animals. They're not a worm or anything like that. They're not an invertebrate like some people that don't know too much biology might think. Limbless, of course, they lack eyelids, so they have that constant stare, but they actually do go to sleep and do things like that. They're what is referred to as a gape-limited predator. So anything that they can stretch their mouth around, they can swallow. But if it's too big to do that, they can't. And so this actually is the case. It's a very large gape that's possible. So something like a rattlesnake has a skull bone design and elastic skin that allows for their mouth to be opened tremendously large. And vipers have been shown to swallow things that are up to 100% or more of their overall mass in size. And so this is a single meal. And a colleague, again, has suggested this is similar to us taking something like a 70-pound hamburger, eating it whole, and not having limbs or any other structures like that to try to manipulate it. So here's a European viper eating a fairly good-sized rodent. You can see the skin is stretched quite a bit there. So what this allows for, though, is snakes can be infrequent feeders. They have a massive meal, and then they can go for a long time. And by a long time, I mean a long time. It's been calculated that a prairie rattlesnake living on the eastern plains of Colorado only needs the equivalent of 2 mice a year to survive. Typically, they're going to feed more than that because they are opportunistic and they actually actively hunt. But they also have extremely efficient digestion. And this allows them to reach numbers that are very high density. And so this is a picture similar to one that shows up online sometimes, but I took this picture at a den site where they're using this culvert as a retreat. Now, unfortunately, for the last couple of years, we haven't seen a huge number like this. Some of the locals know about this, and some of the people who I would consider to be less educated go out there and kill them en masse with shotguns and other things like that. So they're strongly persecuted. Well, if we look at snakes globally and we look at them from a variety of different perspectives, what we see is that there's a fascinating diversity of species that are found globally. The family Viperidae is shown here, and these are the vipers. They include a variety of things, both large vipers like a gaboon viper found in tropical Africa to smaller vipers like this Ph.D. viper found in parts of China. The elapids include things like cobras, coral snakes, and a variety of other things, the sea snakes. This is a sea snake taken on an picture taken on an island off the shore of Taiwan. And then there's a group of snakes referred to as the colubrids that are what are called rear fang snakes. And that's not meaning that the snakes are, or that the fangs are in the back of the snake, but they're in the back of the mouth. And so we've done a lot of work with these, and I'll just briefly touch on these. In Colorado, the only venomous snakes that we have are rattlesnakes, and we have 3 different species: prairie rattlesnake, the midget-faded rattlesnake, and the Massasauga. Prairie rattlesnakes broadly distributed, this one in the western slope area of Colorado, and then this one in the southeastern portion of Colorado. And we've worked with and continue to work with all 3 of these. In your neck of the woods in Oregon, we have 2 different species, and again, they are distributionally limited. The northern Pacific rattlesnake, quite variable, can be found all in the western part of the state, in the more mesic areas. And then over in the drier areas, deserty areas of Great Basin, in the eastern part, we have the Great Basin rattlesnake that can be fairly common in some areas. So you have 2 rattlesnake species, again, for both Colorado and Oregon. If it doesn't have a rattle, it's not a venomous snake. Well, venoms are not limited just to snakes, but there are lizards referred to as the Gila monster here. It's found in southwestern United States and down into Mexico. And then the beaded lizard here that's found in northern Mexico all the way down into Central America. And they have a very different situation. The gland is in the lower jaw. It's associated with grooved teeth, and they can, in fact, have a bulldog bite. It turns out that their venom typically is not very toxic, but it's an extremely painful bite that they can give. Now, just last year, we had a guy in Denver that had 2 of them as pets. He wasn't being very careful, was bitten by one, and held on for 4 minutes, envenomated him, and he ended up dying. And this is the first case of a fatal Gila monster bite known since about 1930. So a very unusual situation. Part of this is because most people don't let them hang on you. If you can, you rip it off your hand, even if you open up a big wound. But the longer it's in contact, the longer it can transfer venom. Snakes have a very different sort of system, and it's a very much more efficient system, especially with the vipers. They have a long fang associated with a large venom gland. They can strike and release very, very rapidly, and so they can deliver a large amount of venom and then get out of the way almost instantly. Cobras, kraits, and their allies, as shown in these pictures here, have a much shorter fang, but they tend to bite and hold on. And their venom, though, contains components that are very rapid acting. The rear fang polypids have an even different system. It's associated with a different kind of gland. It doesn't have a muscle sheath to compress it, but it's passively compressed when they crunch their jaw muscles down. It's delivered to the back of the mouth. This is the maxillary bones, so the front here, the back here. And associated with that are enlarged, sometimes grooved teeth that conduct the venom into a wound that is made. So it's a rather different system here. For the most part, these don't represent risk to humans, but there are 5 so far known that have, in fact, fatally envenomated humans. So we do want to be careful with some of these. So, basic question, what is a venom? It turns out that most of it is proteins and peptides. And this is, when people ask, is this a poison or something? And I said, no, it's as natural as milk. Milk has a lot of protein in it. You wouldn't necessarily want to inject it, but you could, in fact, consume venom. And there are some kind of, I don't know what, less illustrative people that do, in fact, as part of a show, extract venom and then drink it. It has the consistency of egg yolk, so I can't imagine it being particularly interesting to drink. But the point is that it actually has to be injected. And so this allows me to be able to extract venom, get some venom on my hands, wash it off without being envenomated or have any deleterious effects. And that contrasts with what we call poison, which is typically consumed. Well, this venom contains a whole bunch of different biological activities in the forms of these proteins and peptide molecules. And so if you think about the total number of proteins that are out there in the world, there are tens of thousands of them, but only about 10 or 15 are really commonly seen in snake venom. So there's something about those particular proteins that make them good as venom components. what we see also is that they're within a single protein family, and this is a molecule that is called a three-finger toxin. There's actually three of them superimposed here, and they have the same basic structure on a basic chemical level, but they can have very, very different kinds of activities. So even within a single protein family, we get slight differences that have evolved, and this turns out to have very, very different pharmacological properties. And that can make things challenging to deal with as far as treating snakebite, but also makes it very interesting as far as looking at these molecules as a source of potential therapeutics. So there is mutation along particular amino acids, substitutions in some of these loops here. And in fact, that can change something from being essentially non-toxic to being very potently lethal toxic. So we can have the same looking molecule, but very, very different effects. Well, if we think about the evolution of venom toxins, snakes initially, well, they started out a little bit before this, but some of the early snakes were constrictors. They would have to grab prey, wrap their body around it, and then constrict it. And when you're doing that to another animal, the animal typically doesn't care for that. And so you can see this small python has oriented this mouse very properly here so that it can't bite it. But frequently what we see in constrictors are bite marks, that is, scars from their prey. So this has favored the evolution of other types of strategies. And one of the other strategies are chemical-based predation. What we mean here is we can have a viper, which you can just barely see here, that has dispatched prey here. It's crawled after it to relocate it and now swallow it. But it has struck at the prey, envenomated, and gotten out of the way very, very rapidly. This coral snake, on the other hand, is an elapid related to things like the cobras, and it has to hang on to and then swallow its prey, in this case, a small other snake. This coral snake is only about 1 foot long. Well, the interesting thing about venoms is that the genes that code for them started out as normal genes, if you will, kind of normal housekeeping regulatory molecules, take care of things like blood clotting and normal functions in the body of the snake, just like our own. But what has happened is we get duplication of these genes over evolutionary time. They're sequestered in a specialized gland called the venom gland, overexpressed, and then we have basically either some of these give rise to something that's dysfunctional and they typically are selected against. Others undergo what is referred to as neo-functionalization. Because they're now removed from the selective pressures favoring doing things a certain way, they're now stuck in a venom gland. They can evolve all over the place. What has happened is that this is what's giving rise to those protein families, a single protein family with lots and lots of different types of gene copies within that, each one of those varying individually. And so that's what gives us a wide variety of molecules that look the same, but actually do very different sort of things in the venom. Okay. Well, one of the things I like about working with venoms and venomous snakes is it allows me to exercise my ADD tendencies and do a variety of different types of subdisciplines within biology. So comparative anatomy, evolution ecology, protein chemistry, proteomics, transcriptomics, genomics, all of these are central to studying venoms. We spend a lot of our time on the protein end of things because this is where the metal or the rubber meets the pavement, I guess we should say, where these are the molecules that are actually doing the work that venoms have evolved to do, which is to stop prey. First step, we extract venom from them. If they're rear-fanged snakes like these two here, we have to give them a 2-drug dosing to first immobilize them and then cause the cells to secrete the venom. For front-fanged snakes, such as a crate here, we can actually put these capillary tubes on their fangs and then pressurize it. And for the big vipers, we can actually produce a lot of venom by compressing that gland. And my thumb here and my middle finger on the other side are doing that. And you can see there's a bolus of venom here, which is probably 0.5 milliliter or so of venom. So vipers can produce a lot of venom. Once we do that, we can subject it to a couple of analyses, and this is gel electrophoresis. It's a very simple technique where we can separate molecules based on their size. So the large molecules are up here, and the small molecules are down here. And then we can put a whole bunch of different venom samples across here in each one of these, what are referred to as lanes. And by looking across here, we can very rapidly get a good idea of what is in the venom and what is not in the venom, because we've done a lot of work with these venoms, and other people, of course, have as well. So we can basically just do this simple picture here and say, oh, we've got a whole bunch of these molecules here, which are snake venom metalloproteinases. And we've got a bunch here in this one, the big thick bands, very little here, almost none here, same almost none in here. So this allows us to compare venoms from very different snakes very rapidly. And it's the kind of experiment that undergraduates who don't have very much experience can do, get essentially publishable results very, very quickly. Okay. All right, so here's a list you'll probably want to copy down on your hand or something just for quick reference, maybe not. Anyhow, if we think about all the different proteins that are in venoms, there are somewhere between 20 to 30 or so venom proteins, but there are some that are much more prominent. And so the ones that are underlined here are the most common. As far as the ones that are causing pathologies in humans, it's even a smaller subset of that. And so it's these 4 on the left here, as well as this myotoxin, the specialized one that we find in some of the rattlesnake venoms. And so we'll talk a little bit more about these. So if we think about putting down prey, how are you going to do that chemically now, and not physically grabbing it, but instead injecting it with a chemical, in this case, venom? Well, the neuromuscular system is a great place to target, because this is, in fact, how movement is controlled. And so nerve impulses come down, they're transmitted into a specialized region of a muscle, and then that is telling that muscle to contract. Turns out that that is highly regulated with a whole bunch of different types of channels. Some of these are what are referred to as voltage-gated channels, and they're sensitive to the flux of charged ions back and forth. And so we can have things that happen presynaptically and postsynaptically. The synapse is the gap here, and so here's the neuron coming down, presynaptic sites, all sorts of different ones. If we look at who's doing what with snakes, there are some very specific ones found in the rattlesnake venoms that are presynaptic toxins, and they have these 2 subunits that make up these molecules, and this is where we find these in some of the rattlesnakes, some of the vipers. Some of the elapids found in Australia, the really, really toxic ones, also have neurotoxins that are presynaptic that are based on these same kind of structures. At the postsynaptic site, a favorite place to hit is, in fact, the acetylcholine receptor, because this is when a nerve says contract, it's sending a chemical signal from the nerve terminus down to this motor end plate, and it's acetylcholine. And this is the specific receptor for that. So if you block it with these 3-finger toxins, that's going to result in knocking out that whole transmission. And so what we get are things like fasciculations, where you get these kind of weird contractions of the muscle leading to flaccid paralysis. And this can be lethal, because a lot of these toxins localize in our breathing apparatus at the phrenic nerve diaphragm junction. They can also be very, very prevalent in a particular venom. Up to 40% might be one of these molecules or one of these molecules in a venom. So that's going to cause that venom to be very, very toxic to anything that is susceptible. Well, we can also target the muscular system, and we just talked about some of the toxins that hit the neuromuscular junction, but these are specifically hitting muscle tissue. And one that we've got in our area is a little one called myotoxin A that can be very, very abundant in some of the rattlesnakes, and this causes a tetanic paralysis. So as the neurotoxins causes flaccid paralysis, instead, the myotoxin A and chrotamine produces tetanic. And so a mouse that's envenomated by a rattlesnake with a lot of this in their venom show this rapid immobilization, all tensed up. Their legs are hyperextended and very tense. Well, vipers in particular, but some of the elapids and a lot of the colubrids also target the vascular system. And there are a lot of different molecules that do this. Some of them are what are referred to as snake venom metalloproteinases. Some of these are phospholipase A2s. These actually destroy cell membranes themselves. Some of them are more specific in their action. And so they liberate little molecules in the blood. One called bradykinin is one that when it's liberated into the blood, it causes vasodilation, blood pressure falls, and it can fall very, very rapidly. The guy that was killed by the Gila monster had a major issue with this situation there. We have some that interfere with platelet functions, and platelets are these little aggregates of enzymes that help your blood to clot in the right place at the right time. Some of them promote aggregation, some inhibit aggregation, so a bunch of different things that can have activities there. One of the ones that we see commonly in the rattlesnakes in particular are these that are destroying fibrinogen. And fibrinogen is one of the first steps at producing a molecular patch to keep your blood from leaving out of a blood vessel. So if you have snake venom metalloproteases leading to breaking down of the blood vessels themselves, followed by incapacitating the capacity to clot, we've got a double whammy situation. There are a couple of the other ones that are found in weird snakes like the boomslang that cause activation of some of the blood clotting factors that can cause you basically to have very, very severe coagulopathies. And these can be fatal even in the case of this rear-fanged snake, the boomslang. Okay. So we've got a lot of different sites that can be targeted. And in many cases, these regulatory sites in our bodies and the body of prey are hit simultaneously. We're fortunate to be much larger than something like a mouse. And so the dose that we get relative to a mouse is much lower, but it can still be very, very problematic. Well, one of the things that keeps me up at night is how do snakes house this stuff in their venom gland for long periods of time? So what's involved with the production of venom and the storage of venom? Well, if we want to tell the snake to go ahead and start producing more venom, we can do this fairly easily. Extracting the venom manually here from this fertilance then sets into gear a whole bunch of different types of things. The parasympathetic nervous system is stimulated. We get pressure release that's sensed and says, okay, start kicking things into gear. The cells themselves increase in height by 50 to 100%. They're turning into protein-producing factories. They're packaging the venom in these little vesicles. That is then going to be secreted across the membrane of the cell itself and filling up the venom. And if you look at these particles here, these are actually coagulated venom proteins. The cells then have returned back to a resting state. And this is scanning electron micrograph of the ductwork within the venom gland itself. And in life, this would be filled with venom. So we have this whole process that's going on. And in the end, we have a happy snake with full venom glands here. Well, it turns out the regulation of this is extremely complex and involves a lot of different factors. And so this is where our work with colleagues down in Texas that are the genomic wizards has really come to fruition, because we're looking at this process in a lot more detail. And I won't go through all of these interconnections, but suffice it to say that snakes have a pretty complex system for going from a full venom gland, empty venom gland, back to full. So in the meantime, though, if they're not being extracted by us or they're using it in prey, they're keeping that venom for long periods of time. And that venom contains these proteins as well as toxins, some of which are enzymatic. They actually have catalytic activity. And these can be stored, especially in captivity, for months or perhaps even years. And if you think of any kind of protein solution in storage, it tends to basically degrade very, very rapidly. So further, there are enzymes in the venom that can self-destruct. And so they can digest themselves and they can digest other components in the venom. So if we think about a rattlesnake venom gland here, and this is a sagittal section showing the whole apparatus, all this gray material here is in between all of these little bands of secretory epithelium. This is all venom in these ductules. And again, it can sit there in a liquid state for months to years and still be ready to go at a moment's notice. In particular, our attention was turned to the snake venom metalloproteinases because these chop up other proteins. And so they help, potentially they can help digest prey as the snake has taken it in. They can lead to breakdown of capillaries and hemorrhage. So they're very problematic. In a snake bite, these actually cause very severe hemorrhaging. They can lead to tissue damage, tissue death, necrosis, and they can lead to loss of something like a limb. Well, our perspective is, how do the snakes do this? So how are they actually keeping these things ready to go without destroying themselves? Well, storing it in inactive form is the way that they do this. And it turns out that there are a number of different ways to do that, and it has to be stable enough that it's not hurting the snake, but not so stable that it's turning off the venom completely. We started looking at some basic aspects of how the venom is stored. And it turns out the venom is stored under acid conditions. And if you look at where most of the enzyme toxins are active, that's what these bell-shaped curves are, versus pH here, what we see is that at the storage pH, which is about 5.5, all of these things are essentially turned off. And so we have a situation then where the venom itself is acidified. We were wondering how, in fact, that occurs. It turns out there are little cells called mitochondria-rich cells that are found in the venom gland itself. They're not secreting venom proteins, but instead they're secreting acid. And this is very much like what we see in the glands that are found in our own stomachs and the stomachs of snakes. And these are parietal cells that are secreting acid because our stomach is actually digesting food under acid conditions. Well, this is one way that snakes are keeping things under wraps, and that is the pH, or the acid base level, is very low. But because this is a fairly serious scenario, there are backup systems. And one of the things we also see are some peptide inhibitors. And these are small amino acid chains that are about 3 amino acids in length that are relatively low affinity inhibitors. And so they are at high concentration in the glands, and they're not real good. And so when the snake injects, these are going to diffuse away. And so what we have then are a couple of different ways that the snakes are able to store their venom in a ready-to-go form, but not digest their own tissues. And that one of these is storing at acid pH. So these inactivate things. The snake venom metalloproteinases are notorious for chopping themselves up as we try to purify and characterize them. Then we have the problem of how do we turn these back on? Well, we don't have to worry about that. There are 2 log steps up towards neutrality when venom is injected into tissues of prey or tissues of humans. And so it's spontaneously activated. Because we have those peptides are going to diffuse away as well. And so, in fact, we have a very nice system with at least 2, maybe 3 different mechanisms that are storing the venom in a patent but ready to go fashion. So we have these cells that are very much like cells that we see in the stomach, but they're utilizing a different kind of mechanism for producing and secreting acid. And so I have one of my graduate students has been working on this and looking at specifically how do they control this and what kind of effects does it have on venom if we actually artificially increase the pH. Well, this already mentioned that venoms also can vary in overall biochemical composition geographically. And so depending on where you are in the world, we can have a venom with very, very different kinds of components. One of these is that myotoxin A that causes that tetanic paralysis. And in our area of Colorado, there are very, very high levels of this. We started looking at this range wide, and it turns out that in southern populations, it's very low, and in northern populations, it's very high. And it turns out also this is a moderately toxic, it's not super lethal compared to cobra venom, but it's fairly toxic component to mice, has absolutely no effect on lizards at all. So we've got a specific type of toxin that causes hyperextension, tetanic paralysis. And when a mouse is envenomated by these snakes, it's stopped almost instantaneously. If we look at southern snakes, they have a very, very different type of venom with different types of components in there. And this myotoxin A, modeled here, is basically absent in the southernmost ones. This has some practical applications because, in fact, the effects on prey are different, but the effects on human are different as well. And so the antivenom that we use to treat the bite may, in fact, need to be tailored geographically as well. So one of my graduate students, Kara Smith, completed her Ph.D. a couple of years ago, and she was looking at the range of prairie rattlesnakes from right almost at the Canadian border all the way down to the Mexican border. And then all these dots show you the sampling here. It turns out if you know where the I-70 corridor is in Colorado, the difference between the northern and the southern venoms falls right about on the I-70 corridor. We don't think that's a barrier per se, but it is interesting, and it's a obvious geographic feature that people can localize. So we get this changeover very, very rapidly, and it looks sort of gradual, but then all of a sudden, right here, we get a changeover from the northern to the southern and the southern to the northern phenotypes. And so what we have is a very fast-acting toxin-rich venom in the north and a digestive type of venom in the south. The basic question is, why is this geographically so different? Well, we think it has to do, in fact, with the fact that snakes are feeding on prey like this kangaroo rat here. And it turns out that snakes are not the only ones that have evolved over time. Rodents have a very short generation time, and so their evolutionary responses could be even faster than the snakes. And so what we see is that there's resistance that occurs between some of these prey populations, and then that puts selection on the snakes to diversify their armor of different types of toxin proteins. And of course, that changes the selective regime for the rodents and back and forth. And right now, it looks like the snakes that produce this myotoxin A are ahead of the game. And they, in fact, are very, very potent against prey like kangaroo rats, whereas the southern phenotype venom is nowhere near as toxic to the same animals. Okay. Well, another study that I just want to mention is how do snakes utilize this chemically mediated predation to their advantage? Again, they're feeding without limbs, so they're under some different types of challenges. And we're going to look specifically at vipers, and that includes things like the golden eyelash viper here from Costa Rica. And this is a rattlesnake that's actually, both of these are in a predatory mode, waiting for something to come by. And they're very, very patient, and they may be in this ambush mode for days, waiting for something to come by. Okay. They strike, envenomate, release in about a quarter of a second. So the snake avoids retaliation from prey that wasn't too keen on getting bitten. But the prey can wander off to a fairly significant distance. And so, we're interested in how do they actually do that? How do they find their prey after it's been envenomated? Well, a colleague has shown that there is a phenomenon called strike-induced chemosensory searching. And so they're using those chemical receptors, that tongue flicking, that sense of smell and taste, to relocate their prey. And they can differentiate a track from an envenomated prey and a non-envenomated prey very, very exquisitely. So if we think about this picture, if you think about a snake sitting here and a mouse approaches, snake strikes, and the mouse can run off in a variety of different sort of directions, maybe even go underground in a burrow or something like that. So there is a chemical trail that's been set here by the mouse that's been envenomated. But the mouse may have been wandering all around the area before it encountered the snake, and so there are chemical trails of a non-envenomated mouse all over the place. There may be other species trails going hither and yon as well. And further, it might be nighttime, and so visual cues are going to be completely absent. So the snake has a problem. Which track is it going to follow here? Well, David Chazar, who was a colleague that passed away a number of years ago, did a lot of work with this strike-induced chemosensory searching, and many, many different species show this phenomenon, but it is most developed in the vipers. We asked the basic question, what in the venom is allowing them to do this? And we thought, okay, there's a lot of enzymes, things that digest tissue, that's probably going to be the target. And so David developed a behavioral assay that we utilized as well. And long story short, we took the venom apart into large, medium, and small molecules using a technique called size exclusion chromatography. And we used those, each one of those peaks, as it were, to test for, can the snake relocate the prey if it's been envenomated not from the whole venom, but from venom fractions, as we call it. It turns out that none of the ones that we thought were in fact the case. There was a little peak here. We purified that, sequenced it, and it turns out it is a molecule called the disintegrin. A disintegrin has a very interesting property, but it's not toxic. And so you could actually give this at fairly high doses. And in fact, it's not going to cause death or incapacitation of the prey. So that was very curious to us. So we've got a situation where we have strike and release envenomation. This gets the snake out of the way of the prey, can't bite it if it's not there, but it does in fact create a situation where we have a prey that has wandered off somewhere. We may not be able to see it at all, and so we have to relocate it. This is seen only among vipers, and those disintegrins are found only in viper venoms. And it turns out that when we injected these into prey alone, that allowed the snakes to find their envenomated prey. So it's been wondered for a bit why we have a non-toxic toxin in a venom. And it turns out that the reason we have that in there is, in fact, this allows them to relocate their prey. As again, these are found only in the viper venoms, and vipers commonly are sit and wait predators. They wait for something to go by, they strike it, it runs off, dies somewhere else, and then they have to relocate. All right, so one of the subtitles of this talk was Toxins to Therapeutics. And it turns out that reptile venoms are pretty good sources for actual and potential therapeutic compounds. But if you think about it, it doesn't make sense a priori, because venoms evolved with a very different sort of function, not to treat cancers, but instead of killing to kill or immobilize prey. But if you remember back when we first were talking about those evolution of those compounds, they're not just some de novo poison. Instead, what we have are these regulatory compounds that are found in the body of vertebrate animals in general that keep things regulated under a variety of different types of conditions. And so if you think about a mammal in particular, talk about a very high-tuned system. So this is like a race car that works best under just so conditions. And so many aspects of physiology, things like blood pressure, blood sugar, oxygen levels, acid-base balance, things like that, are very highly regulated. And if we fall out of kilter in that, we start feeling pretty bad, right? Well, these toxins are whacked out regulatory compounds that the snake's injecting at extremely high levels relative to what the normal regulatory compound would be. And it's at an inappropriate time and place. And so essentially what happens is all the different regulatory systems are affected very, very rapidly, and overall chaos, or in this case, mouse death, occurs very, very quickly. So again, humans are larger, so we don't typically have that immediate response unless it's a weird situation like a envenomation is right into a major blood vessel, vein in particular. Okay. So these different molecules, though, affect systems in a variety of different sorts of ways. Some of them are more lethal, some of them are less lethal. And we can think of this as evolutionary tinkering. And so over millennia, hundreds of millennia, we have, in fact, all sorts of different variants that are of toxins that are out there in the natural world. Now, if we take a crude venom, we're going to have the effect of basically a shotgun blast. And I use the analogy that you could take a tumor out from a human with a shotgun blast, but you'd get a lot of collateral damage. So we take the venom apart, purify it, and if we have desirable effects, then we may be able to use that therapeutically. Some of you, like me, may be on an ACE inhibitor. And in fact, this was first based on a toxin found from the South American viper. And this was back in 1965. It's been refined a bit, but captopril is still being used and is basically based just on that molecule itself. Tirofiban, agrostat, is from another viper, saw-scaled viper. This is actually one of those disintegrin molecules. Remember that they are found in venoms of vipers, but they're not toxic. Another one here is a serine proteinase that actually is, in fact, being used as a surgical glue because it can, in fact, produce adhesive conditions in things like fibrinogen, but it can be metabolized away. There's another disintegrin here that is used for treating coronary syndromes and heart attack and angina from a pygmy rattlesnake, something that's found in places like the southeastern US. And then the Gila monster. Again, we had a bad rap for the one that killed the guy last year, but in fact, there are a number of different drugs that have been derived from Gila monster venom. One of these has been marketed as exenatide or Byetta and is pretty good for controlling type 2 diabetes. And there are another set of these that are now being marketed as Ozempic Wegovy from Gila monster venom as well. Very closely related to these molecules, but they have in fact been modified slightly. And Novo Nordisk in Denmark is a company that has marketed this, and it has been a tremendous blockbuster drug. And so all of these are from reptile venoms, most from snake venoms, but the big blockbusters, other than the ACE inhibitors, are from Gila monsters. So there's good evidence for thinking that these might be a good source. I'm going to run through this a little bit quickly, just because we're getting a little bit late on time here. Integrants have a lot of potential. Turns out they interact, or disintegrants, I mean, interact with cell molecules called integrins. And these I refer to as the chemical legs of cells. They actually allow things like metastatic movement of cancer cells. And so one of my students isolated one of these from one of the rattlesnakes, and this is a big rattlesnake from the Yucatan in Mexico, and showed that it actually was not toxic, but it in fact inhibited migration of melanoma and cancer cells. And it does this in a very specific sort of fashion. And it's not killing the cancer cells, but in fact, it may allow us to use lower doses of standard chemotherapeutics. And if we can use those to kill cells that are localized, that may lessen the side effects. And if you've known anyone that undergoes chemotherapy, it's a rather severe therapy to undergo. So we have a situation where this molecule has in fact evolved for prey relocation by the snake. We have been able to repurpose it therapeutically, and it has anti-metastatic properties, anti-cancer properties. And so here's a situation where we have basic research that can inform aspects of applied research. And so there's really this interaction between them that's critically important. We have worked on a number of different molecules. And again, I'm just going to go through these quickly. Some from the rear fang snakes that have anti-parasite properties. Some from the vipers and elapids. Some of them that act as anti-clotting agents, so we can transiently keep your blood from clotting if you're undergoing surgery or something like that, and then reverse that by stopping the treatment. We talked about this disintegrin from the rattlesnake venom. And then more recently, one of the snakes from Australia, the king brown snake, which is a lethal snake, has in fact some compounds in there that seem to have both antibacterial property and wound healing promoting properties. Okay. So I'm going to just pass over this. We have a lot of different properties, or a lot of different new therapies that are in fact being developed that are allowing this movement from the bench to the bedside, if you will. And I can talk about these if you'd like a little bit later, but again, we're getting late on time and I don't want to keep you too long here. So if we think about venoms and venomous snakes, we have a system that's fairly complex. involves a lot of interesting aspects of gene regulation, neural regulation. We can work with them on the whole organism level, all the way down to the individual gene level. And in fact, many of our new drugs are proteins. So if we understand how snakes stabilize these for up to years at room temperature and higher, then in fact, that might give us models for stabilizing our own drugs. We've seen how venoms can vary in composition depending on where you are geographically, and that can be problematic as far as treatment. We see how snakes and their venoms allow for this hands-off feeding, as it were. And so hopefully you've gotten the idea that venoms are really a rich source of biological materials that are useful for drug therapy and drug discovery. And so if you think about this in a broader context across the globe, there are tens of thousands of venomous species. Many of them are spiders, scorpions, and things like these marine snails. All of these produce venoms for obtaining prey, and all of them are potential sources for therapeutics that we can turn then to use for human disorders. Well, this work has been done in collaboration with a lot of different people all over the world. This is my friend in Singapore. And of course, graduate students working in my lab have been fundamental and central to a lot of the work that we've talked about here today and things that are ongoing as well. We've had great relationships with landowners who actually don't kill rattlesnakes in their property. And of course, we've had funding from a number of different sources, which is critically important to take the science to any kind of level that we would like to. So with that, I'll thank you for your attention.
So this is a piece of information and a question.
Okay.
Okay, kingfishers are birds,
Yeah.
and they were highly endemic to an island in Guam. Okay,
Yeah.
until the birds fell victim to the invasive brown tree snake,
Yeah.
accidentally introduced to the island in the 1940s. Now, how often do snakes intrude on avian species?
Well, there are a number of different species like the brown tree snake that are adapted for feeding on birds and lizards. And it's interesting you bring that one up. I didn't really mention it, but we worked with the venom there. I went to Guam a couple of times, had permits to get 18 snakes the first time, and in the first 3 days I was there, we got 43 snakes. So they're very, very common still, even on Guam. This was back in 2001 when we first started. But brown tree snakes are arboreal. They can get good size. So they have led to cataclysmic declines in birds in Guam in particular, because it's a very small island. Nothing was preying on the brown tree snakes. As they knocked the bird population down, approaching zero, they started shifting over to introduce black rats. Those started to crash a bit. They also ate some of the large fruit-eating bats that were native to Guam. It really caused problems with that population. Most problematic for humans was they caused over 1,000 island-wide outages because they crawl up on the power lines and then bridge the transformers themselves. It, of course, electrocutes the snakes, but it also shorts out the whole grid. So lots of problems associated with them. So brown tree snakes in their native habitat have a variety of different predators, and so they still feed on birds. And a lot of different arboreal snakes, like this viper here, in my background here, feeds on birds as well. It's Costa Rican. So we've got a lot of them that feed on birds. There's an island species found off of Brazil that is almost entirely dependent on birds. But if we go back to the brown tree snake for a moment, it turns out their venom is not particularly toxic to mammals like us, but it's incredibly lethal to lizards and birds. And there's a neurotoxin found in their venom that is as potent as the cobra neurotoxin, but only to lizards and birds, and it's essentially harmless to mammals. So that led us to come up with this idea of a taxon-specific toxin. And we found this in a lot of these rear-fanged snakes, and the brown tree snake is one of the rear-fanged snakes. So predation on birds is pretty common. A lot of species like rattlesnakes are more terrestrial, but they will in fact feed on ground nesters like doves and some of the other species that nest on the ground. Occasionally they'll make forays up into shrubs and trees and such, but generally speaking, they're more terrestrial. So,
Well, thank you. And now it's not only
Yeah.
not only the cats that are doing a job on the birds, but we have to worry about snakes as well.
well, in a specialized thing like the island, a small habitat like that, birds are particularly vulnerable. There's been a lot of work trying to keep them off of the islands of Hawaii. And there have been at least 7 that have been found on airport tarmacs and destroyed.
Wow. Wow.
So no evidence that they've gotten established there, but it's hard to believe that before the late 70s, some of them didn't get from Guam to Hawaii because there was a lot of traffic, air traffic and ship traffic.
Right.
So,
yeah, thank you.
Mhm.
I appreciate it.
Okay, let's go to our number 1 here at Friend.
Oh, that's right. We don't have to go to the microphone. When I was a boy, I was taught the following. Red and yellow killed a fella, red and black, friend of Jack. But you were showing a venomous snake that was red and black and white. So what?
Yeah. Well, that rhyme works really well in parts of the United States. There are some of the coral snakes proper are found from Arizona over to Florida. The one in Arizona is a small one, only gets about a foot long, so it's not a problem for humans. But the ones from Texas to Florida are large enough that they could actually kill a human if they envenomated. Most of those, they have the red and black venom lac, red and yellow kill a fellow. They follow that little rhyme. But there are variants in those populations where they don't follow that. Now, once you get into Mexico, all bets are off. And as you go from Mexico to South America, there's a great diversity of different coral snakes, and they could care less about that rhyme. They show all sorts of different patterns. There's some that are blue and black patterned. So again, it works reasonably well in the United States, but south of the border, no.
I grew up in Texas. Water, but I guess there was more.
Okay, I think we have a quick question here. Is the venom gland salivary gland?
It's a form of modified salivary gland. So yeah, it's obviously very different. If you even ourselves, we have a number of different salivary glands in our oral region. Snakes have even more, because if you think about it, swallowing something whole that's kind of dry and kind of rough surfaced requires a lot of lubrication to get it all the way down to the stomach. And so what has happened is that a subset of those glands, one gland, has evolved to produce not only mucus, but proteins. And those proteins, turns out, are actually fairly toxic now. So probably early on in the evolution of snakes, they were not particularly toxic. But over evolutionary time, and snakes, venomous snakes have been around for somewhere between 50 and 110 million years, depending on the lineage. So there's been a lot of evolutionary time for those venoms to diversify, and that's what we see. So,
okay, let's go to question number 2.
The vipers and rattlesnakes, I think all of them have a pretty obviously wide skulls, like the one in that yellow one that you in your photo. Yeah, compared to cobras or I think most non-poisonous snakes. So what's the function of that wide skull?
well, it turns out that the lower jaw joint is all the way back. It's actually about 30% of the skull length behind the end of the skull. So their jaws extend backwards really far. What that means is the lower jaw is really long, and then there's a bone called the quadrate that articulates with the skull proper, and that kind of flares out sideways. So that whole triangular look is because those bones are elongated in this particular line of snakes compared to other lines of snakes. Practically, what that does is it allows for the snakes to have a very large gape. And so if vipers got the size of boas and pythons, some of the big giant snakes, they could easily swallow humans without much, you know, a 12-footer could easily swallow humans, but vipers don't get that big. They still can stretch, they can drop their lower jaw very far down. The tips of the mandible, the lower jaw, are held together with ligaments so they can actually separate and expand out laterally like that. And then, of course, the skin of the snake can expand tremendously, about 2 to 3 times what it would be in a non-expanded state. And so what that allows for then is, remember that snakes are gape-limited predators. So if their mouth is only that big at a max, that's the biggest prey it can take. Well, now this allows for that gape to be that much bigger. And so that's why vipers, they've actually, a colleague has found a viper that had a prey in its body, it was digesting it, that was 150% of its overall body mass. So 1.5 times the size of the snake itself, which is pretty amazing.
I'm sure I'm going to have marvelous dreams tonight.
So,
can we go to number 3 here?
I would like to go back to color for a minute. Every snake, almost every color, every snake that you pictured had a number of bright colors or at least a variability in pattern. And I wonder what is the advantage of all that color. I don't know what the chief predator of a snake would be, but I should think you want the color to blend in with the surroundings, but these stand out.
well, I mean, part of the selection is based on the fact that, you know, these are pretty colors, right? So they're a little more attractive to an audience and things like that. There are a lot of snakes that are fairly dull patterned, and those are much more cryptic. So, for example, the Massasauga rattlesnake, it's a little rattlesnake. It only gets about 15 inches long or so. And we did radiotelemetry with that. That is implanting an actual radio in the body. And then we can use a receiver and antenna, and we pick up a little beep, and we can relocate the snake on a repeatable basis all through its active season. So we can follow it around, see what it's doing. We had one snake that was radioed. We knew it was around a base of a bush in about 1 meter diameter area that we were getting the signal. And it took us about 10 minutes to see the snake, even though it was sitting on the surface. So that pattern of blotches and dark light really blends in very well with the background substrate. Now, some of these snakes, like coral snakes and some of the milk snakes and king snakes, have these red, black, and white bands that are very, very obvious. And they stick out like a sore thumb. You'd think, well, what's the advantage of that? Well, that was kind of answered to me one time in the hills of Southern California, where I found one of these mountain king snakes, red, black, and white banded. It was on oak leaf litter. And when it moved, it just kind of disappeared. It's what we refer to as a disruptive pattern. So it just kind of, you know, it was much harder to see the snake than when it was sitting still. So part of the adaptive significance of some of these patterns may be this disruptive aspect. There are some that have very long stripes down their body. And if you think about a predator going after something, it's focusing in on a point. But if that point is moving and it looks the same up by the neck as it does almost to the base of the tail, the predator can't really tell that this has gone by until it's too late and the snake's gone. So there are a number of different features that seem to influence the patterns. Really cool one is that there is a snake in Central America that the first part of it, the first half of it, is banded like a coral snake with that red, black, and white pattern. and the back half of it looks like you took another snake and stuck them together, and it's just brown. And so it may be kind of a blend of both, that we've got this warning coloration that's saying to a predator when it's out on the surface partway that it, I'm mimicking a dangerous snake. It's not a venomous snake. But then when it's foraging and its back end is out here, more vulnerable, it's very, very cryptic with leaf litter and things like that. Does that make sense?
To Rich Kronostowski on Zoom.
Yes, hi. I was wondering, obviously the snakes are limbless vertebrates. So
Yeah.
when evolutionarily did they lose their limbs, or did they simply never have limbs? Or they're a very early lineage that simply never developed limbs?
There's actually a paper published on that in Nature a number of years ago, on the news article published in the same journal said, "Nice snake skeleton, too bad about the legs." That was the title, right? But basically what it was is I want to say about 120 to 150 million year old fossil that did have remnant legs, both fore and hind limbs. Now, if you look at modern day boas and pythons, in particular in the males, there is a remnant pelvic girdle and a tiny pelvic limb that's referred to as a spur. And it's not involved in locomotion. I mean, it's, on a 10-foot long python, the spur might be a 1/4 inch long. So it's not involved in locomotion at all. It's exaggerated in size in the male, and the male will, while he's courting a female, put his body over the female, and he actually can move his spur like that. It's like he's giving a little love tickle. And that may, in fact, either induce receptiveness or let the female know what his intentions are, and she'll take off. So the limbs have, snakes in general have evolved from lizard-like ancestors. And it's been a long time now, 150 plus million years, as we saw that transition. But there actually are some fossils of various lineages that have limbs, albeit small. So
I couldn't.
How about number 4 here at Friendly Hat?
The bifurcated tongue is very, prominent. Is it the bifurcation mostly a surface area, or is it for actual direction finding somehow, or wind and things like that?
there's been some debate about that. But there was a paper that was published a number of years ago that pretty convincingly showed it was a detector, left-right detector. So if you think about that fork, right? If you have a scent line that goes straight up and down like this, as you start to veer off to this side, you're going to get differential stimulation on this side, and that allows the snake to correct. And as you go off to the other side, this one is differentially stimulated, and so that allows it to correct. And the effect is that it can do this kind of movement along a straight line. So a number of years ago, we were working with, I think it was Discovery Channel, on a program, and we did this scent tracking experiment in the field with a snake out in the field. And we laid down a rodent track on one and just a water track on the other. And it was pretty amazing, because I was pretty convinced that there's no way this is going to work. But that snake followed the trail of the scent just right to the money, right? Straight line right to where that scented, in this case, it was just a scented sponge, where it was. Producers like, well, that was good, but we need to get some more shots of this again. So we repeated the whole thing, and I'm thinking, come on, having this work once is pretty amazing. Did the same thing. Snake again followed the trail of the scent 100% accurately. Well, that's pretty good, but we'd really like to. So we did a third time. The snake all 3 times did it with a high degree of fidelity. So it's pretty impressive.
If the snake swallowed the sponge, that could save a doctor some work. Number 5 here.
Yes, the traditional antidote for snake bites is ethyl alcohol. Do you have any evidence of any interesting interactions between ethyl alcohol and venom in humans or other animals? And are there any perhaps medical applications for this?
Well, if you take venom and pour it into ethanol, you can denature the proteins. But when you've been bitten by something like a rattlesnake, those venom proteins are now injected in your tissues. And you might be able to get a little bit off of the surface by pouring ethanol on it. But as far as drinking ethanol, that's contraindicated by almost everybody in the medical profession. It's going to increase the probability of circulatory distribution of the venom proteins through your body. That can lead to more systemic rather than localized effects. And those are the kind of things that can kill you more rapidly. So generally speaking, if you have been drinking, and strangely enough, there seems to be a correlation between consumption of alcohol and incidence of snakebite. In particular, among a certain group of humans, males, typically Caucasian, between the ages of 18. So.
But wasn't it recognized by the U.S. government as an exception to prohibition?
I'm not sure
that came up in the discussions, but maybe you have some information on that.
We have a question on Zoom that we kind of touched on. Can any snake swallow an entire human?
Again, that's the kind of thing that we see pictures online, and some of these are obvious fakes where the person says, this giant python found in South America, and it's like giant pythons are not even found on that side of the world. So sorry. So there's a lot of fake stuff out there. There are some diminutive individuals in some native populations. I think it's on one of the Philippines islands that a colleague actually thinks there's very good evidence that several of those individuals over hundreds, maybe thousands of years actually have fallen prey to big reticulated pythons. And by big, we're talking about something that can get 20 to 30 feet in length. And so the diameter of something like that can approximate this. So what is that, 18 inches or so? So there seems like there's a possibility that humans could be consumed by those pythons. If it's still happening, it's extremely rare. Very large boas and pythons are very rare these days in general because people kill them either for food or just to get rid of them or to use their skin as leather products. So there's a possibility, but it's probably not very likely that humans are eaten by snakes.
I think we have time for a couple more. Number 6 here.
West of Syracuse, New York, there's a national wildlife refuge, and there's a Cicero bog. And we were told as students that that was the Mackessy was the only poisonous snake in that area of New York State. Is that a different species? Are you familiar with that species?
Yeah. That's one that's been reevaluated recently as well, as far as its affinities with different species. Used to be the desert Massasauga that we have in Colorado was considered the same species as the one that's found in New York and up into Ontario. Now they're considered to be two distinct species. And the eastern Massasauga is a considerably larger snake as an adult compared to the Colorado snakes, which are the desert Massasaugas. As adults, they're probably about a third smaller at least as far as length, but as far as overall mass, they're a lot smaller. So yeah, the eastern Massasauga though is in big trouble. It's been extirpated, just like a timber rattlesnake, almost everywhere it used to occur. But the eastern Massasauga also was in wetter lands that largely were drained for building and stuff like that. So it has kind of a 1-2 punch against it.
Thank you. And we have a question 7 here.
Yeah, I spent a little time in academia, and while I was there, somebody said to me, you know, academia is just filled with weirdos. And I want to thank you for demonstrating that's true in the most wonderful way.
No problem. We always I refer to academia as a retreat for socially inept individuals. So.
Real quick one.
That you take that snakes have to be coiled to strike, and then they can coil quickly. Any comments about that?
Snakes can kind of swing their body side to side pretty quickly. They're much more efficient at striking if they're from a coil, and that's what they typically coil up like that in a ambush coil or a predatory coil, because then they can launch their body and hit something that's a little bit farther away. But you know, if a snake is fully stretched out and you were to put your hand within 2 or 3 inches of its head, it could strike laterally without too much trouble there. Wouldn't necessarily be inclined to do so, but it could. Yeah.
This is wonderful. I think we can all take this information and be really popular at cocktail parties.
There you go. Yeah.
Thank you again. This was great.